Okay, assignment this Friday. No begging for extensions, it's got to be on Friday because I'm outsourcing this marking because I can't cope with all the marking. Sub story. But if I can't hand them off to this person in one big batch, you miss out. So if it's not in on Friday, what's that? You can, yeah. You got the money to pay for it, no problems at all. So make sure it's on Friday. So today's part 2 of anaerobes, part 2 of wounds rather, talking about anaerobes today. So we can get a bit too fancy about how to define anaerobes, but I'll try and keep it really simple. I used to go into how they respire and what the chemical formulas were for respiration, and I don't bother with that anymore. It's a bit too theoretical and not so practical. In the end, think of anaerobes as bacteria that either won't grow or won't survive in the presence of oxygen. That's a fairly simple definition. And if you look at all the bugs that we deal with medically, there's a whole spectrum of how they tolerate oxygen. So some bugs are aerobic, strictly aerobic, like Pseudomonas, and they can't grow without oxygen. They need oxygen for the end point of the respiration, and they make energy. And other bugs are facultative, and they can do either aerobic or anaerobic. What was that? Something important that just on the floor? Phone? An umbrella, okay. With the bugs that we call anaerobes, these are the ones that won't grow under normal conditions of anaerobic atmospheric conditions that we would find in the lab. But even within the anaerobic spectrum, there's a whole range of how much they don't like oxygen. So there are some anaerobes that are aerotolerant. What this means is they won't grow in oxygen, but they can handle it if it's just not growing. They'll just sit there, they won't die, but they won't multiply either. And then amongst the ones we call obligate anaerobes, these are the ones that simply won't survive if there's oxygen there. Won't survive or grow. Some of these are very strictly obligately anaerobic. This is getting really complicated. These ones need to have less than 0.5% oxygen in their atmosphere. And there are some that are moderately strictly anaerobic. They can handle about 2 or 3%. For us, it doesn't matter because we can easily achieve about 1 or 2% oxygen in most conditions. So we're good at growing and handling these kinds of anaerobes, the ones that are moderate. We're not very good at the ones that are super strictly anaerobic. There are lots of bugs that are truly anaerobic that we can't culture with normal procedures. You've got to be really fancy and really careful to grow those, and labs that do that are specialised. And that's particularly important with mouth microbiology or gut micro. We can grow Bacteroides and all those other things simply, but there are so many anaerobes in your gut you can't grow because they just die like that when you're exposed to oxygen. So we're going to deal with mostly moderately obligate anaerobes. They won't grow in anything more than 1 or 2% oxygen. There's a whole range of reasons why they can't handle oxygen. In the end, as long as you understand that when they metabolise in the presence of oxygen, they make toxic byproducts. We don't want to know the chemistry of it all. It's a bit too complicated, even for me, so I'm not going to bother with it. In the end, just to understand, most of the bugs that we call anaerobes need to have less than 1 or 2% oxygen, and we're happy with that. I'm going to try and go through them in a way that's systematic, just to try and get your head around them. So, of the gram-negative anaerobes, there's only one that's a gram-negative coccus. It's called Veillonella. And if you look at a chart that shows you how often we grow certain anaerobes or report them from medical specimens, Veillonella is hardly ever seen. Could be a whole lot of reasons. Could be it's not common, full stop. Could be that it's common but we can't grow it very well. Could be that it's common, we grow it and we can't identify it. Either way, it's not commonly reported. So, for us, end of the story. But the gram-negative bacilli anaerobes are really important because they're so common. We can think of them in terms of how they respond to three key antibiotics that are useful for identification. So these drugs here, we aren't talking about whether you can use these for therapy. It's about high-level discs used to separate bugs into three different genera. Well, four. So we have the Fusobacterium. These are all gram-negative rods. Fusobacterium, it's vancomycin resistant but susceptible to kanamycin and colistin. Now these three discs are three of the discs in those anaerobe ID discs that we used in the lab the other day. They're convenient, quick tools to use to separate gram-negative anaerobes into these four genera. And it's helpful. What we want to have are tools that are cheap and quick that do a reasonable job. Then there's Bacteroides fragilis. We call it one species, but in fact there's a whole group that belong to that species. We only ever just call it B. fragilis. And they resist all those three drugs. And Prevotella and Porphyromonas used to be in the same genus as Bacteroides, but they were changed because A, these can be pigmented, and they're also bile sensitive. So Bacteroides is bile resistant. So you can make media that's selective for Bacteroides and against the other gram-negative anaerobes by including bile in your media. Because Bacteroides can tolerate bile, and these don't tolerate bile at a high concentration. I mean, obviously if they're bowel flora, they can handle bile, but we're talking about a high level of bile. So Prevotella and Porphyromonas, these are just gram-negative rods. In a gram stain, these three look identical. In a gram stain, Fusobacteria are quite different. They're normally very distinctive. So it wouldn't be hard for me to say to you, in the practice exam, for example, you should be able to easily recognise and identify those four anaerobes. Not hard to do. First task is detect it, prove it's an anaerobe, do a gram stain, do those discs, give it a genus name. So the top diagram is a, will you tell me what that is? Oh, I'm too scared to do the amplify. Where's the, what do I do again? Shift and plus, is that right? Control and plus, thanks. There you go, like that. So what are they? What kind of gram-negative rods are they? Are they boring normal rods? Are they long, thin rods? Are they needle-shaped rods? They're needle-shaped rods, which puts them in what genus of gram-negative anaerobe? Fusobacterium. It's not bloody hard, is it? Pretty easy, really. A Bacteroides wouldn't be like that, nor the Prevotella, nor the Porphyromonas. And these ones there are the ones we saw in the prac, the bizarre shapes of either Fusobacterium necrophorum or mortiferum. So if your gram-negative rod anaerobe is a boring, old, pathetic, simple gram-negative rod, it's probably Bacteroides, Porphyromonas or Prevotella. If it's something a bit more interesting, it's probably something like a Fusobacterium. Control and the minus button, like that. Oh. Something about doing gram stains of pus that's fantastic. Now, this is a gram stain of some pus, and when you see that gram stain, once you've done this for a while, you straight away say to yourself, this will grow anaerobes. Because you're seeing a whole range. So this is a simple wound swab on somebody's arm. You wouldn't see all this sort of stuff. You might see some staphs, for example, but all these long bacilli here, they're anaerobic. And over here, this is a Fusobacterium. All these smaller rods throughout there are mixed anaerobes. This could have come from any of a number of kinds of sources. This could be a tooth socket in your mouth. So you've had a tooth out and it's gone all impacted and sore and throbbing away. Do a swab of that and you're bound to get mixed anaerobes. It's the kind of wound you're going to get it in. If you had a deep wound in your arm, you wouldn't get this sort of picture. I've also seen this from under a foreskin that was infected, the same kind of thing. Lots of anaerobes because of the nature of the body site. Could be a high pelvic swab, similar sort of thing. So you tend to see typical things in terms of the ecology of an anaerobic infection. And now in your mind, you've already got a map that shows you all the gram-negative anaerobes. Here's the gram-positive anaerobes. Of the gram-pos cocci, the most common are the peptostreptococci. Let me turn the lights back on again, sorry. In that genus are two species, Anaerobius and Magnus. They're the two most common. There's also a strep called Strep milleri, which is microaerophilic. What does that mean? If you're microaerophilic, you're not truly anaerobic, but you won't grow in the amount of oxygen in a normal CO2 jar. So a microaerophile likes extra CO2, but oxygen a lot less than ambient oxygen. How much oxygen is there in normal air? About 20%? These grow at about 5%, happy at about 5%. Which other bug do you know that's microaerophilic? And it was quite recently you encountered it while I was away. You read about it. Campylobacter. We use the same gas pack to make an atmosphere for Campylobacter as we do for this one. Again, my memory, it's yeah, that's right, CIA. In the group Strep milleri, you'll see how it's got inverted commas there. Milleri is no longer an accepted name for a species, it's a group. And because it's a long-term used sort of name, we still like it. But it really consists of 3 different species. Constellatus, oh, it should be small c, Intermedius, and Anginosus. And these streps are what I call small colony type streps. So on a plate grown next to group A strep, they're much smaller, they're tiny. And they can have hemolysis, beta hemolysis, but not necessarily. If you group them with a Lancefield group, they can group as A or C or G or F. They're a bit weird. And we often think they're anaerobes because they won't grow in normal air, but they're not truly anaerobic. The good thing is if you make a mistake and think they're anaerobes, it doesn't matter because they cause diseases in the same place as anaerobes because they like an atmosphere of not much oxygen. They're common causes of deep abscess sort of infections. You won't grow them from the skin. It'll be a deep wound somewhere, often internal, an abscess, liver abscess, or an abdominal mass that you suck the pus out and you grow something, a brain abscess even. The great thing about them is they smell beautifully. They smell like caramel. In the prac, I will guarantee you, I will give you the treat of smelling a lovely caramel organism. It's great. When you get one, it's like petrol sniffing. You just can't get enough of it. You just carry the paper around for the whole day and keep giving it a sniff. It's lovely. So bear in mind, these are not true anaerobes. Of the gram-pos bacilli, there's the spore formers and the non-spore formers. Non-spore formers, actinomyces as a genus name. Actinomyces are long, gram-positive branching rods, and their normal habitat is mucous membranes, say in the genital tract and in the mouth, some in the bowel, but more mouth and genital tract than anywhere else. And they are very, very anaerobic. They die pretty easily if you're not really careful. Then there's Propionibacterium. Propionibacterium, small pleomorphic gram-positive rods, a bit like Corynebacterium, but the anaerobic version. And these are predominantly skin flora, but not on the skin where all the air is, deep down in the crypts of your hair follicles. And they're linked to cystic acne because when the hair follicles get blocked with fats and oils, these bugs overgrow to form pimples and pus-producing lesions, so that's what you get really bad acne. So the most common species here is Propionibacterium acnes. The reason it's important is not because it causes disease, it's a very uncommon cause of disease, but because it's skin flora, it's common contaminant of a range of kinds of things we culture. It's a slow grower. So imagine you've got a pussy wound in your arm, Staph aureus, and Group A strep. You're lucky you got both. And even an Aeromonas at the same time, all three of them. You've been out playing in the surf and you've hit your arm on an oyster or something and you've got an infected. If I culture your swab, blood agar, McConkey, MSA, the whole lot, in 2 days' time, I've grown the Aeromonas, grown the Staph, grown the Group A strep. I'm happy as Larry. I've got a good result out of this swab. At 48 hours, I'm done. It's all finished, gone in the bin. The risk of a Propionibacterium skin flora being a contaminant that gets in the way of my culture is zero because it's a slow grower. It takes a week to grow. So we don't see this often as a contaminant in specimens that have pathogens. But one of the things we do a lot of is blood culturing for unknown sepsis. You've got unknown temperature, you can't work out what's wrong with you. You might have some underlying low-grade septicemia, or you might have a lesion on your heart. We take blood and we culture it in a broth for 10 days before we chuck it away. That's when we get growth of Propionibacterium, not as a pathogen, but as a contaminant from when we do the blood collection. It's important use, if you ever see a Propionibacterium in a blood culture bottle, you think about what that might mean. It's nearly always a contaminant. Except this can also cause long-term slow infections of the heart valve. So sometimes it's a pathogen in blood culture, other times it's not. I'm jumping the gun now, but while we're on the story, we might as well keep going with it. I wonder how you'd know whether what you grow as a Propionibacterium from a blood culture is a pathogen or a contaminant. Because remember, you've taken this blood out of someone's arm, 10 mils, put it in a broth, incubated, and you've waited for 24 hours, 36 hours, 48, on it goes. At some point before 10 days, the bottle goes positive by some mechanism. You know it's gone cloudy, or a bell's gone off on your machine. What makes you think that the growth in one circumstance is a pathogen and the growth in different circumstances is not a pathogen? No, because most are negative. 95% of blood cultures are negative. So negative is always going to happen. And you almost never get two bugs in the blood culture. So what else might make you think it's positive? This is probably an unfair question to ask you. We'll get there eventually. The amount. So with a blood culture, it's like this. When you put the blood in the bottle of broth, in the syringe is an X amount of organism. It might be 1 cell every mil, might be 100 every mil. And you put it in that little bottle. And then you put the bottle in an incubator and they grow, and some kind of system detects the growth. How fast your system goes from neg to pos depends on the starting number of bugs in your broth. If you start with more, it goes positive earlier. Start with less, goes positive later. So it makes sense. If you're infected and there's lots of bug in your blood, then your syringe full's got hundreds of bugs in there, goes pos faster. Contamination might be 4 or 5 bugs or 10 bugs, goes positive later. So how soon it goes pos from collection and incubation is an indicator of the starting inoculum, which is an indicator of most likely disease or most likely not disease. That works only if your system of detection is really, really good, and most times it's not. So it is helpful, depending on where you work and what your machine is like. Something else is helpful, and you probably don't know this yet. When you do blood cultures, you take more than one collection. So you take one sample now, you go away, and you come back in 4 hours' time, another sample. You go away, another 4 hours' time, another sample. If you truly are infected, you expect that this sample and that sample and that sample are positive. Contamination is more likely to be only one of those three positive. So the number of samples in your set that are positive is really helpful. We'll go over that again in another 2 weeks' time. So don't stress about it. So these are non-spore formers. Of the spore formers, there's Clostridium. Clostridium is a variable organism. The most common one, Clostridium, is a thick, brick-shaped, gram-positive rod with big, fat spores. And the spore location can change, but it's normally either central or subterminal. And when you gram stain them, if they're old, you see a hole in the gram stain where the spore would be because the spore isn't stained. Some of the other Clostridia that we don't grow so well, like tetani, is a whole different shape. I wonder why we have a hard time growing Clostridium tetani. It's one of those left-hand side anaerobes that's really, really touchy about oxygen. And if you aren't careful how you manipulate your specimens and your plates, they die. So they're really hard to grow. And they're slow growers, and the colonies are flat and pathetic, therefore you miss them quite easily. Clostridiums, you can find that with glasses on, sunglasses on, I mean. They're so big and massive, you can't miss them. Clostridia sometimes stain gram-negative, and there are some species of Clostridium that are always gram-negative, but their cell wall is gram-positive chemically, but they don't hold the stain very well, and they stain like gram-negative rods. Right, what kind of anaerobe is that? That's an actinomyces. Long, gram-positive branching rods, and they often stain inconsistently, so they don't stain lovely and purple, they're spotty almost. One of the few times you can grow these, when you grow these from some specimens, they're eaten with so much normal flora, and they grow so slowly you can't grow them. So in the vagina, for example, they do grow and cause disease, but they're really hard to get out because when you do the culture, all the coliforms and enterococci and the bacteroides grow so much quickly that these get obscured. But there's one body site where you tend to see them occasionally, but it's pretty cool when you see them. There's a thing called dacrocystitis. And dacrocystitis, memory's right, is a blocked tear duct that's infected with actinomyces. And because that sample would have almost no normal flora, you've got time to grow these things. It will take about 7 days for a colony to appear with actinomyces. And that's a Clostridium. Big, fat, gram-positive rods with holes where the spores are. Subterminal spore, terminal spore. Control. It's a blood culture. So you come in the morning and you've gone to your blood culture machine and you've looked at the chart on the front and it's gone, bottle 25, red light flashing, it's positive. Pull the bottle out, take some broth out of the bottle, onto a slide, do a gram stain, what's going on? And you see that. These are small, pleomorphic, gram-positive rods. And these are red cells. And that is almost definitely a Propionibacterium acnes. Sometimes it's important that you can deal with things quickly and not waste time on them. Wouldn't it be a shame if you spent days and days and days agonising over what this bug is? The gram stain is quite distinctive. It would only go anaerobically, and they're Spottingdale positive. The Spottingdale test is one of those things that can be really useful in 3 or 4 situations, but don't overdo it. If I see you doing spot indoles in the prac exam on bugs that don't deserve them, bad karma. You know, the guaranteed way to fail the exam immediately is to do a spot indole on a staph, just because you can do one. What's the point? Say to yourself, if I do a spot indole on this bug and the answer is pos, what does it tell me? If the answer is neg, what does it tell me? If the answer is the same for pos or neg, don't do one. Only if you can get some information would you do one. So E. coli, do one for sure, because it's useful. A bug that you think might be Pasteurella, do one because they're spot indole positive and it's a really useful test. As other stuff, you know, don't waste your time. And that's tetanus. That image was taken in about 1989 at the LGH, and we didn't grow anything. It was a negative culture, but it was there, and the fact was we just weren't good enough at keeping our specimen anaerobic to grow them. They're really, really hard to grow. You might have one in the collection. I might try and grow one for you. How good is your eyesight? Probably better than mine. So, I want you to have a picture in your mind about what kinds of body sites contain what kind of anaerobes. And anaerobes tend to live on mucous membranes, because mucous membranes maintain a very low tension of oxygen. They're anaerobic. Your gums are really anaerobic. All the saliva around your gums keeps those things really anaerobic. Vagina, very anaerobic. Bowel, very anaerobic. Gut, very anaerobic. But we have different anaerobes in different spots. And as a rule of thumb, I draw a line about there somewhere. And for those who aren't here who are listening to the recording, and I'm saying there somewhere, you can't see what I'm pointing at. Bad luck. I'm pointing at about the throat. So below the throat is where you find Bacteroides fragilis and Clostridium perfringens. So really what we're saying is Clostridium perfringens and B. fragilis are really bowel or genital tract, not oral organisms. In the bowel, for example, there are so many bugs. I mean, enormous numbers. 10 to the 14 per gram of stool is bacteria, and 99% at least of the bowel flora is anaerobic, and 1% is coliforms. E. coli is a terribly small part of your bowel flora. And of those anaerobes in there, only a small proportion of them ever cause disease. Clostridium and Bacteroides most commonly, and also the gram-pos cocci. In the genital tract of females, again, we'll have, what's important there is, even though it's below the neck, we don't get B. fragilis and we don't get C. perfringens. If you have B. fragilis in a genital infection, it's probably come via the bowel. Now, this list here is a list of how commonly we isolate and report anaerobes in specimens in a pathology laboratory. And how common they are reported is a combination of two things. One, how common they are, and two, how easy they are to grow and recognise and identify. Got to be both things together. Some things are really, really common, we just can't grow them, so we never report them. So being common and being easy to pick up is helpful. So the most common anaerobe are the gram-pos cocci anaerobes. They grow pretty well, they're pretty easy to recognise, they grow well on most of our media, and identification is pretty easy. Really, if you just prove that your bug is anaerobic and do a gram stain, that's it. You give it a genus name. Then B. fragilis. One of the reasons for this being so common is A, we have good media that we target this bug, because this bug is quite aggressive, so we target the bug pretty easily. It's common normal flora, and identification is pretty easy. Propionibacterium, commonly reported, but hardly ever a pathogen. Most commonly, it's reported as a contaminant. And then Clostridium, and most of those to be perfringens. And then it drops right off below that into Prevotella, Porphyromonas, and Fusobacterium. So as long as you're okay with the top 4 of those, you're laughing. It's possible to waste a lot of time looking for anaerobes when you're never going to find one. And it's dispiriting to keep looking and looking and looking and finding nothing. So my message is, don't look for them, except when they're likely. Because you spend resources, the right kind of media, mucking around with all kinds of incubators and atmospheres for no purpose. There would be no point, for example, in looking for anaerobes in urine. You might see a UTI caused by anaerobe once every 1,000 years, so you could spend all that time doing it for no benefit. And the same with things like a skin swab or a simple non-deep wound swab. You won't find anaerobes in an arm wound, for example. Unless your arm's been cut off with a chainsaw, you might, but just a simple arm wound, you're not going to find them. There's no point looking for anaerobes also anywhere where anaerobes are normal flora. A vaginal swab, why would you culture for anaerobes? Because it's normal. So if you cut, you're going to grow them every single time. That's where they live. So what's the point of doing it? So you try and put all your efforts into doing it when it's necessary. So here's some examples of times when anaerobes are known to be frequently involved in infection. And I put a point there saying involving, not causing. And often the anaerobes are not primary pathogens. They come along after something else. They take an opportunity when it's presented to them, but they're not really good at primary causing disease, like Staph aureus and Group A strep are primary wound pathogens. Anaerobes tend not to be. And we nearly always get anaerobes, not always, but most often anaerobes come with other organisms. And one of the reasons is that the other organisms help to maintain an anaerobic environment. The other bugs do 2 things: consume the oxygen and cause tissue damage that makes the whole tissue anaerobic. So when would you see, what other things would you see with anaerobes? What kinds of bugs would you see with anaerobes? In deep wounds, for example. Messy wounds that smell. What would you see with them? Coliforms, ad nauseum. Coliform. Lots of gram-negative rods are facultative. They're facultative, they use all the oxygen. And you find coliforms in the same kind of body types you find anaerobes in the bowel. So you often get anaerobes with coliforms. What else might you find that's a gram-positive that's catalase negative? Strep. Oh, it used to be called strep once. Yeah, enterococci. Again, they're bowel flora. You often find enterococci with the anaerobes. So, aspiration pneumonia, a really uncommon disease, but when you see one, you wouldn't forget one, and they're horrible. It's caused when you lose consciousness and you aspirate either your mouth contents down to your lungs, or you vomit and you swallow the vomit. Imagine, you think about your mouth. Remember that mouth gram stain? You had bugs everywhere. Imagine if somehow that stuff goes from where it's up here, it's normal, into your lung. So when you're unconscious or you're drunk, and you lose control of your swallowing reflex, and you open your epiglottis and it goes down the wrong tube into your lungs, that's horrible. And rather than being a pneumonia like we normally see where it's quite diffuse, this is really an abscess in your lung caused by mouth flora. I wonder which kind of patient group is most at risk of aspiration pneumonia. Looking at Jared. Looking at Gabriel. Young males, yeah, who get on the chirps and get so blind drunk that they either vomit or they pass out and they swallow it. I'm way past the worst sort of boozing period now, but you know, when I was at your age, it was just like, it was a ritual, wasn't it? Is it still the same now? I don't know, but I used to come to work and you go, how was your weekend? Oh, it was great. What did you do? Oh, I got pissed. And you go, oh, I was so pissed. No, it was even worse than that. I was terrible. I was maggoted. It was like a pride. You know, I just can't imagine the amount of money I wasted. All that time wasted doing that for no reason. Stupid. But when you're that drunk, you lose control of stuff and you start to aspirate. Right. Any kind of mouth infection, either a punch injury or a break, and especially a deep tooth extraction. Having your wisdom tooth out, for example, leaves a great big cavity in your mouth, and there's all those bugs there into the tooth cavity causing infection. If you have an infection, say, of a tooth cavity, what's the thing you're scared of? There's something else, like, I don't want these bugs in my gum, because they're not very nice, it would give me an aching mouth. But there's something else close to my gum that I don't want them in. My jawbone, yeah. You don't want to get bacteria in your bone. Bone and bacteria are absolutely a bad mix, because it's really hard to get the drug into your bones. So treating osteomyelitis of any body site is really, really hard, because there's no drug penetration to get into that site. So it can be nasty. Pelvic sepsis, talking about either pelvic inflammatory disease or any kind of sepsis involving the pelvic cavity, normally involves bacteria that have ascended from the vagina or the uterus, and it involves anaerobes. Actinomyces, Prevotella, Porphyromonas. Horrible, hard things to treat, hard to diagnose, because when you're trying to culture them, how do you separate the pathogen from the normal flora? Because they're the same thing in some ways. One bug's taken over and moved up higher to where it's causing disease. So often they're treated empirically. I mean, you get signs of pelvic sepsis, and you might do some scans and some imaging and some blood tests, but you won't culture an organism and say that's the culprit. You'll treat for pelvic sepsis based on probably anaerobes who'd use drugs which are unknown to be good against anaerobes, penicillin, perhaps clindamycin, metronidazole, things that we know are good at anaerobes. Any kind of abscess. What's an abscess? Abscess is an internal, walled-off collection of pus. They can occur for the weirdest reasons, often around your liver, in the abdomen, in the lung, in the brain. An abscess is almost guaranteed to contain an anaerobe. Any kind of abdominal wound. You know, anywhere below here is got risk of contamination with bowel flora. So wounds on the abdomen, wounds on the lower legs become quite deep wounds quite often, and they contain anaerobes. And those wounds contain anaerobes and coliforms and enterococci and pseudomonases. Post-surgery, any kind of intra-abdominal sepsis. You've been sick and you've had a bowel resection. They cut you open, pull part of your bowel out, sewn you up, and there's a leak from where they've sewn your bowel up. Leaks out, next thing you know, it's all swollen and red and angry, and it's full of anaerobes. It's almost impossible to imagine. You know, we've talked a lot about this in the press, about the risk of antibiotic resistance. And we always think about the danger is, oh, I've got an infection that can't be treated. The one thing that's the biggest fear about ongoing resistance to antibiotics is the loss of the protection of prophylactic therapy. The only reason we can do bowel surgery now successfully is before surgery we hit you, and during surgery, hit you with a whole lot of antibiotics. And that means that during surgery and just after, any bugs that are there are knocked off. While you heal, then you're all okay. If you couldn't do that, you couldn't do bowel surgery. So there's a big risk of drug resistance is we can't do lots of things to really have the chance to be prophylactic. And then foot ulcers, either diabetic foot ulcers or pressure wound ulcers. And we talked last week about this. It's when either the pressure of your body stops blood flow or poor circulation from diabetes stops blood flow. In the end, the tissue breaks down because of poor blood supply. Once it breaks down, the tissue breaks down, and then you've got an open wound that will become colonised and or infected with organisms. Staph, coliforms, pseudomonas, enterococci, anaerobes. We'll talk in a minute about a gangrene as a separate thing. Gangrene is a separate kind of infection that usually involves Clostridium perfringens. And we're talking about rapid and massive tissue destruction following infection. And we'll talk about that in a minute quite in depth. This is a pressure wound on somebody's lower back. And I can remember having great fun at a nursing workshop. I used to do like pracs for nurses. It used to be in a workshop, not actually in the lab. And I had this long scenario about this lady who went in the hospital and got sick and blah, blah, blah. Went on and on and on. And I told them this was an old lady. And I told them that she was on vancomycin. And I told them that vancomycin had a side effect to give you a hairy backside. And they all believed me. It's not a lady, obviously. But you can imagine there you'd have enterococci, you'd have coliforms, you'd have pseudomonas, and you'd have anaerobes. When you culture that, it's really hard because what do you target with your culture? And what do you look for? Do you look at every single bug and pull them out and isolate them, give them a name, test drug sensitivity? Or do you report as heavy mixed growth, including the following organisms? And there's no simple answer. It depends on where you work. The best labs are pragmatic about this. You can spend a lot of time. So in there, what if you grow 5 bugs? Staph aureus, Klebsiella, Enterococcus, Pseudomonas, and the Bacteroides. By the time you pull those 5 bugs out and purify them, and identify them, and do drug sensitivity testing on those, probably 4 days of work in that, and a lot of money, and it won't change anything. You're better off saying heavy growth, including these organisms, and the clinician will say, well, I think based on looking at the wound, this needs to be treated, and I'll treat for the primary pathogen in that group. But you can get too carried away in reporting everything, and you'll find that a lot of labs now have become so scared about doing the wrong thing. They report everything and become really slow in the process. We don't think it's a bad thing. So you can see now, this is bowel surgery gone wrong. So there's the surgical wound, and it's really red. What do you call that redness? Cellulitis, and it's swollen. It's bumping out. It's pushing out. This is gangrene, and the swelling is the gas inside, and the gas is normally from Clostridium perfringens. So here's a quick summary of how we get gangrene. Starts off with an injury, and the injury causes a wound. If in that wound you have implantation of spores of C. perfringens. Now it may be from the soil, because C. perfringens is common in the soil. It may be from your bowel, but somewhere the spores must be there. The injury itself causes tissue damage and makes the tissue become slightly anaerobic. And then we have other bacteria there that are facultative, that grow and make the wound even more anaerobic. So overall, a combination of the injury and the bugs that are facultative making this anaerobic causes the wound, the spores in the wound to germinate. Once they germinate, there is no faster bug that I've ever seen in growing than C. perfringens. It will grow from nothing in a broth to being bubbly and cloudy in 4 hours. It's really, really quick. So as they multiply really quickly, they are strong gas producers. And they make gas. And as the gas comes out of the organism when it's growing, the gas wants to go to the atmosphere. So it bubbles up through your muscle in the shortest route possible and therefore tears muscle on the way up. So the gas production causes more tissue destruction, which makes the whole thing more anaerobic, which stimulates more germination and more growth. There's a circle there of destruction and more growth, destruction and more growth. As well as the tissue being destroyed by the organism and its toxins and the gas, the toxins also spread systemically and yet systemic toxemia. So overall, as a disease, it's horrible and it's rapid. Two things you've got to do, well, three things you can do with gangrene. Treat the organism. Cut off the dead tissue, because once the tissue is dead, has no blood supply, and you can't get the drug in there anyway, and it's dead, so cut it off and stop the circle of more toxin production and more gas production. And try, if you can, to increase the amount of oxygen to the tissues. So sometimes with bad gangrene, they put them in a hyperbaric chamber to increase the amount of oxygen getting to the tissues from external sources around the wound. And without that, you know, you're going to lose your leg. Or, yep. No. That wasn't a quick, definite no. Let me think about the reasons why you wouldn't get them in the blood. To form a spore, the bug should be stressed and under nutrient stress. If you're in the blood, you are laughing, mate. You've got stuff everywhere. You don't want to form a spore. You're going to go for it while you can before someone kills you. Forming a spore there would be just bad form for the bug. The only way to get these spores in the blood is by injection. So you can do it that way, but an infection wouldn't turn to a spore in your blood. So when would you get spores in your blood from injection, Jared? When would you get spores in your blood from injection? Bone mite. You know, isn't it funny? So, yeah, no, you're right, foam mites. But when I think of foam mites, I think of things like pens and toothbrushes. So I've got these spores in my toothbrush. How does it get in my blood? What kind of foam mite? Very specific. Needle. Drug users. Dirty heroin. Dirty drugs. Spores. Now that is gangrene. And it's had an amputation, and more than likely, they've got to go back in and redo it because they've not gone far enough. So they cut it off, try to stop it advancing, and now look at all the blisters there. This is gas in there. It's all swollen, lots of cellulitis. Got to go back there, cut some more off. And in this image here, these are all the clostridial cells, and the smooth muscle here should be lovely and compact. All these lines there are broken by gas production from the spores. Not nice. Okay. One more slide, then we'll have a break. In the lab, lots of specimens do not contain anaerobes, and they're not even part of your thought process. But lots and lots of specimens do contain anaerobes that are significant, and we don't find them. And overall, I would say that the amount of resources we put into finding anaerobes in most laboratories is pretty poor. We don't do it very well. And there's a whole lot of reasons why we don't do it very well. The first reason is they're often polymicrobic. It's an anaerobe plus something else plus something else. And it's in our nature to go, I found something, I'll stop now. If you find a Staph aureus, you go, job's done. Even though it might be a Staph plus a Bacteroides. Or you might grow a Staph and a coliform and miss the Bacteroides because you found something and you think you're finished. Also, they're hard to isolate. When things are hard, we tend to want to avoid doing them. I'm the same. I find Milo really hard, so I just avoid it. I just go, in my life it doesn't exist. I just refuse, it's just hard. If I wasn't such a sook, I'd learn to use it properly, and it'd be great, but I can't. It's hard, I don't like it, so I've just done with it. Same with anaerobes. They're hard work, and if you can find a way to make your work easier, you tend to do it that way, so we often miss anaerobes for that reason. And they're slow. If things are slow, it slows down the whole thing. Isn't that annoying? Here you are with this Staph aureus day one. You want to report it and get it done. You might wait 2 more days to grow Bacteroides fragilis. You're holding up the primary pathogen reporting by 2 days to grow a bug which might not even be important. So if things are slow in micro, we tend to avoid them because we're already a bit irrelevant because we're too slow anyway, let alone being more slow to find anaerobes. One of the major reasons why we do want to grow our bacteria from wounds is, or from any kind of specimen, is to test them for drug sensitivity. The whole reason we want to grow an E. coli from a urine is not to call it E. coli, because no one really cares. It's to test it for drug sensitivity and choose the right antibiotic. If tomorrow they invented a drug that would cure all UTIs forever and never have a problem, straight away, no more urine cultures. That's the primary reason we do it. We don't care for any other reason. Because we can't, or we're not very good at testing anaerobe sensitivity, we do all this work to grow them and can't do the important bit. So again, it's become a bit less than useful. There are no good disk diffusion tests for anaerobe sensitivity. This doesn't work very well. So even now, if we grow anaerobes, we prescribe antibiotics empirically. We say, well, normally a B. fragilis responds to this, this and this, but not this. We treat them on prior information, not on a test result from that laboratory. They grow slowly, and all of our systems we've already developed require fast growth and growth in broth that's aerobic. Because it's a small part, it's almost like no one's taken the time to develop a method that's good for doing them. At the moment, all we can do for anaerobes that's really useful is E-tests. We can put an E-test on a plate and get an MIC, but this diffusion works badly, well, poorly for anaerobes. What's that?
Even something like perfringens, considering you said already that it multiplies really quickly.
The thing with perfringens is, again, it's predictable. So this is the issue down here. It's all quite predictable. If I grow perfringens, the drug choice is dictated by history, not by a test. How else can I explain this? So, coliforms, staphs in particular, Pseudomonas, enterococci, all have really unpredictable sensitivities. You can't tell from this isolate and that one what it's going to respond to, because they've picked up either mutations or acquired genes, and they're quite fluid in how they respond to drugs. For some reason, anaerobes are not like that. It's quite predictable what they're going to respond to. And we tend to use a blanket of drugs for anaerobic infections. Well, not just one drug, there'll be 2 or 3 drugs, certainly 2 drugs to cover a whole range. So in the end, the site of infection, the gram stain that you see from the specimen, all of those things are predictable. So we can get away without being good at culturing. Doesn't mean we shouldn't try, but I'm just saying once you get it as a cop-out, there's a cop-out. If tomorrow I was told you are no longer allowed to email your students, Tristram, that's it, done. What would I do? I'd learn Milo. Because as a cop-out, email, I go email because it's easier. Same with this. If this stopped being the truth anymore and you couldn't predict anything, we'd be better at doing anaerobes. The reason we can get away with it is there's another way out. It's easier. Okay, it's not working. It's 5 to 10. How about we start again at 5 past? 10-minute break. And pause recording. Okay. The key thing to maximising how often you can grow anaerobes is to minimise the exposure of the organism and the specimen to oxygen. And it might sound pretty simple, but it's much harder than you think. Deep wound somewhere. Here's my swab. In it goes through the big sinus hole in your chest. Beautiful, comes out, covered in pus. Straight away, it's gone from in there, anaerobic, on my swab, in the air, aerobic, they're dying straight away. So from specimen collection to culture, that's the key time to minimise the time you expose them to oxygen. So if we think about specimens in general for culturing anaerobes, the first part of that little spiel there should be select the specimens that you put F into and ignore the ones that are useless. So don't waste your time looking for anaerobes in specimens where anaerobes are unlikely or are normal flora. So don't look for anaerobes in vaginal swabs. They'll be there, but they don't mean anything. Don't look for anaerobes in skin swabs. If you find one, good on you, that's great, but don't put resources into looking for them. And don't look for anaerobes in throat swabs. It's all normal. When I say don't look for anaerobes, it does not mean you shouldn't put up a plate anaerobically, because we know that having a plate anaerobically helps us decipher what we're growing aerobically. Because we go, oh, it grew there, didn't go there, therefore it's this, or therefore it's something else. A blood agar plate grown anaerobically is not the same as looking for anaerobes. If your question is, do I have anaerobes in my specimen? Oh, I have looked by doing a blood anaerobically and looking at that. That doesn't work. This is a poor way to look for anaerobes. It won't work almost all the time. It's just one tool we use in general culture to help decipher what's going on. So once you've decided this specimen is one where anaerobes might be important, what you do now is you put resources in that specimen. You make sure it's collected and transported properly, and you use specialised media. Don't be afraid to reject the specimen. So, good example is during surgery, deep wound somewhere, someone takes a swab of this big pus in someone's abscess and puts the swab in a little carrier, swab carrier, that's just air with no media in it. Well, straight away, the swab is sitting in air. Anaerobes are dying all the time. It's drying out. Bugs are dying all the time. Culturing a dry swab is a complete waste of time. Every time you culture a dry swab, you reinforce in the mind of the person that took the swab, that's okay. What you should do is straight away come to the lab, you take the swab, you phone the clinician and you say, sorry, this swab is not acceptable, we're not culturing it. And next time that person will make sure they do it properly, which means transport it in the right way. That's easy to do if you work in a hospital, because the hospital has got a captive population of providers, clinicians that work there, and you can say things to them like, that's not good enough. In private pathology, the whole mindset is different. The mindset is our providers, our GPs, you never argue with them, because if you do, they'll go somewhere else. It's true, isn't it? The whole mindset is we train from day one. Don't argue with them, don't tell them anything, be nice to them. They're complete suckholes, just so you get their specimens. So how much you can give them feedback about their specimen quality is a bit different. But when you can, try and be educational. Say, look, this specimen We'll do it this time, but it would be much better if you did it this way, and you'll get a better result that way. The best way is a needle aspiration. So if there's pus in an abscess, there's two ways to do this. Put a swab in, send the swab. Or put a needle in, suck the pus out, send the whole syringe over, because now there's a syringe full of liquid that's still anaerobic. Comes to the lab, might take 2 hours. In that 2 hours, it's still anaerobic, it's liquid. That's much better than a dry swab that's set for 2 hours, getting dry and being exposed to oxygen. So specimen quality is really important. If you must do a swab, we have special transport media for the swabs. So here's your swab, cotton swab, and there'll be a special container it comes in. At the bottom of the container is some jelly. It's actually agar. And that's called transport media. And it's designed to do a couple of things. Keeps the specimen moist. Bugs do not like to be drying out. It contains buffers and some glucose. It's not meant to grow the bugs, but keeps them alive. It maintains their viability. And importantly, most of them have a reducing agent. Like thioglycolate or resazurin or something chemically that mops up any spare oxygen in that liquid. The liquid, the jelly-like liquid, is anaerobic. It still supports the survival of things that are aerobic, like pseudomonases, but it means that the anaerobes don't die. And you must use a good transport media. It can't be a dry swab. Okay, so that's the first thing. Specimen collection and transport. Get them from collection to the lab ASAP. If there's involved a delay, make sure that they're maintained at a good temperature, not too warm. Now, when you inoculate your plate, this is where the real crunch comes in. This is where you can make an effort. Here's my swab. It's been transported properly. They've been really careful. It might even be a syringe of pus. Here's my agar plate, blood agar. Here's my syringe. Squirt, streak out. My plate is there in the air. And the plate is blood, and the blood contains oxygen. Straight away, I'm killing my bugs while I'm doing it. And you're there, you streak them out, and you know, you're a beginner, and you're streaking it really slowly, like this, you're bloody agonising over it. And then what do? You put it on your bench, because the jars that we have hold 10 plates. So you might even put it in the jar, the blue jar, the anaerobic jar, and it'll sit there on the bench until it's full of 10. And then you put on the lid and you add your gas pack and it becomes anaerobic. On a slow day, it might sit there in that jar in air for 2 hours. They're dead. Anaerobes by then, they're dead. So you want a way of using a holding jar. So a holding jar is a jar you use before you put them in an anaerobe jar. So it's a smaller jar you put them in. It's got a tube on one side that contains some inert gas. You pump the gas in, the chamber becomes anaerobic temporarily until that's full. Then you transfer those to a big jar and put up anaerobically. And now I've even seen this happening. People get lazy. With a holding jar, it involves putting the bug in there, doing up the lid, turning the gas on, 2 seconds or 10 seconds, turn it off. Oh, it's an effort. What I'll do is I'll leave them in the holding jar without doing the gas business until it's full. And then I'll transfer them. So you're wasting your time. So make sure you inoculate them and put them anaerobic as soon as you can. The trouble is, in reality, we can't afford to have one single plate in a great big jar that holds 10 and put in a gas pack, making it anaerobic. Because it means for 10 specimens over the course of a day, there'd be 10 jars. You want a specimen of room. And each specimen, time you put the gas pack in there, make it anaerobic, is $5. So you've got to try and be careful about how this. So a good way of doing it is to say this. I will put my specimens that are possibly serious anaerobe specimens and I'll batch them. It comes in lunchtime, it's in a syringe, it's fine there. I'll put that aside until the afternoon and do all my things that are anaerobic at one time. You're better off having them sit there in their specimen jar, in their pus, or in the transport media for 3 or 4 hours, and then doing a batch process than you are doing individually. The biggest risk is when they're exposed to oxygen, which is out there, not inside there or in a syringe. So batching makes a lot of sense. It's also possible to pre-reduce your plates. And if you're really serious and you want to have some fun, you should try this. If you put a blood agar plate inside an anaerobic jar and make the jar anaerobic, the plate will change colour. The blood cells in the blood agar go from being a light pink to a deep dark red, because the hemoglobin is now anaerobic hemoglobin. What do you call that? Red hemoglobin, hematology. And if you put the bug onto a pre-reduced plate, there's less shock to the system of the bug. It goes, this is blood, it's great, and it's nice and reduced blood. So when you're serious in the morning, you go in, you take 10 blood agar plates, put them in a jar, and pre-reduce them. Then in the afternoon, when you do a batch of anaerobe culturing, use those plates. It's great. It's even possible, and some labs do this when they're super, super serious, they do what's called a pre-reduced and anaerobically sterilised plate. So their plates are kept totally anaerobic from the time they make them. They even sterilise them anaerobically to make them completely anaerobic. And they store them in the fridge in an anaerobic sealed container. If you're culturing like mouth stuff and you're serious about research, that's the level of effort you'll go to make sure that the bugs don't die. But for what we do, as long as you have maybe things being pre-reduced in a good holding jar and being batched, that'll be good. Big labs these days have an anaerobic chamber. So it's a built-in, it's an incubator with a front that's got glass on it and some hands. You put your hands in there, and on one side is a little door and an airlock, and the whole chamber is anaerobic. So you even do your plating out inside this chamber that's anaerobic. They're really good. If you're serious, they're the best thing, but they're expensive, and if they break down, they're hard to repair, but they're very, very good. Okay, so we can make these things anaerobic quickly, and a chamber's a great thing. Here's a problem now, though, is manipulation. The way that labs work is everything is done in batches. In the morning, I'm on the bench of swabs. So I come in, there's the incubator, open the door, get all my jars out, all my plates with the air out, all my concrete agar plates, all my MSA plates, and I open them all up, put them in big piles. Patient 1, patient 2, patient 3, and I start to read the plates. On a big day of 40 specimens, 40 swabs, start at number 1, and I'm looking at that. Oh yeah, step forward, sensitivity test, do this, do that. It might take me 2 hours to get as far as the last specimen. It's now sat there for 2 hours in the air. All that care I've done up there in the first part of my whole procedure, the bug's now in the air. And sometimes exposure to air at a really critical point is bad, and anaerobes grow slowly. So maybe at 24 hours, they've just got the hang of it. Beautiful, I'm growing on this agar plate, this is great, life is good, I'm growing happily. And you pull them out, stick them on the air, and they die because they're vulnerable, their peak growth period. And the same thing there, here's my plate, lovely anaerobe plate, no growth. I'll reincubate that, get my black pen out, put on a big R on the top saying reincubate, put it there. And then work my way through there, and I've got 10 more reincubates, big pile of 10 plates, an hour before they get reincubated for anaerobic growth. It's no point. So it's better if you keep all your things that are serious anaerobes to one side. Do all your plain vaginal swabs, skin swabs, eye swabs, ear swabs, they're great because they don't have anaerobes. But the ones that you really target as being serious anaerobe ones, do them by themselves. And that way you look at them quickly, do the work, prioritise and reincubate them. Be quick and give them individual care, and that way you grow more anaerobes. I mean, if you tried hard for 12 months, you would probably increase your rate by about double, just by simple little things like that. We do also use pre-enrichment. Ooh. What happened? I think, oh, there you go. It's a bit weird. We can use pre-enrichment. So far, when do you use pre-enrichment in other specimens that we've talked about? The salmonella. We take the poo, put it in a sunlight broth, and we pre-enrich for salmonella, and then we plate out that broth the following day. Gives it more salmonella growth. Same with anaerobes. Sometimes they're so sparse in the specimen or so weak that we can't recover them. But if we pre-enrich first and then at 24 hours sub the broth onto some more plates, we have better success. And there are two commonly used broths for enrichment. One is called a cooked meat broth, and all that really is chopped up ox heart in some broth. And in the bottle, it's like this. There'll be a whole pot of broth that's there. At the bottom, big chunks of ox heart. And in there, it's very anaerobic. And they're great things to play with because they smell so nice. When you get them and you incubate them, it's like soup. It's like a broth. Beautiful. The meat's nice. I buy them now. We used to make them ourselves when I first started in the lab. We used to buy an ox heart from the butcher and chop it up into bits and cook it and make our own broth. It was great. So you use that, or use thioglycolate broth. Now, thioglycolate broth, it's a broth that's quite thick, so it's about 0.5% agar, and it's not solid, but when you tip it up, it moves slowly. And it contains an indicator, and it's heavily reduced, so that it should be green when it's anaerobic and pink when it's aerobic. If you take the broth and you shake it, it goes pink because you put all the air through it. But it will come back and recover to go back to being green. And they're good because you can tell if your specimen has become aerobic. So either of those things are good. What you do, swab comes in, primary plates, plus the broth. Next day you come in, if your plates already are full of growth, staphs and coliforms, there is no point then culturing your broth, because you know the broth will be full what you've already grown. So 24 hours, if the growth is already lots, chuck away the broth. If there's no growth or not much growth, then you sub the broth on some more plates. Get a second chance to grow something nice. That's very common. But all of this is great, but the one thing that's most important is this. You need plates that are selective and differential to find the anaerobes, because they normally present as a mixture of anaerobes and something else. So now I'll talk about the kinds of plates that we can use. There's a chamber that's anaerobic. There's one at the LGH that's still in use, I think. And all you do is this whole thing in there is anaerobic. There's the airlock. Everything in there, even when you do your gram stains, it's all anaerobic. The plates there from inoculation to deadification are never exposed to air. And this will really give you a much greater chance of growing anaerobes. Don't get too involved with this quickly, but how do we make our jar become anaerobic? There's more than one way. So a simple way is to buy the gas in a big cylinder. And it will be a mixture of carbon dioxide and nitrogen. Every bug, every single bug that we ever grow likes extra CO2. So it'll be no oxygen, but it'll be CO2 plus nitrogen. And you have a jar with two holes, an in hole and an out hole, and you pump the gas in, it pushes all the air out, the jar's now anaerobic. They're quite cheap. You can buy the gas cylinders, and it's quite efficient to do it that way. It's a bit tedious to do, but it still works pretty well. Most people use a simple little jar, and you add a sachet that makes that jar's atmosphere anaerobic. There's two ways to do this. I don't know now whether the old method is still used by anybody. But I'll tell you what about it anyway. It may be that in some countries you still use this method. In this method, you had a sachet that you used to add water to. And the sachet contained a chemical that when you added water to it, would give off hydrogen gas. So you add the water, it would bubble. You better hear it go. And the hydrogen gas, with a catalyst, would combine with oxygen to make hydrogen, to make water. And that's how you got rid of oxygen out of the atmosphere in your jar. It worked okay. It became anaerobic quite quickly. But you required a very good catalyst to catalyse this reaction between hydrogen and the oxygen. If the catalyst failed, didn't work, the problem was your jar became full of hydrogen gas. And when you open the jar in the morning, out come the hydrogen gas, and you're in a lab with Bunsen burners, and they were dangerous. And they failed quite a bit because the catalysts used to go off. So they've been replaced now by a system, I don't even know how it works, but you buy this little sachet, you tear off the top, you put it in the jar, and somehow it sucks in all the oxygen from the atmosphere, and it's anaerobic. And these never fail, except if the lid doesn't seal properly, then they'll fail. So all of our jars over there are quite old, so we have to use Vaseline on all our seals to make sure they stick properly. When you open them, you get it on your hands, it's horrible. These are much better. They're faster in terms of how soon they become anaerobic, and they don't fail as often. How do you make sure your jar is anaerobic, or was anaerobic? So in the morning you come in and there's a whole pile of specimens you think are going to have anaerobes. Open the jar, no growth, no growth, no growth. Is that because there's no anaerobes there, or because your jar failed and wasn't anaerobic? You want some way of making sure in the morning your jar was anaerobic. Otherwise you can't know what you're doing. So most commonly we use a chemical indicator. And what we use over in the lab, in our lab, is a little pink paper strip. It's a moist pink paper strip. Contains rosazurin. And rosazurin is white when reduced and anaerobic, and pink when it's in oxygen. So in the jar in the morning, if I come in and my little pink bit of paper is pink, I know my jar is not anaerobic. It's failed. And that means my specimen that I read is just not valid anymore. If I come in and it's white, I know it's anaerobic. And the same methylene blue. You have a methylene blue solution in a little jar in your anaerobic chamber. If it's blue, you know the system is not anaerobic. If it's colourless, it's anaerobic. So you want some system to make sure your jar is anaerobic. From the time you put your sachet in the jar to when it becomes anaerobic is about an hour. It'll take a bit longer for the thing to go from pink to being white. So you can't tell straight away. You've got to come back in an hour's time and check. And it's helped to check before you go home. Otherwise, it's too late to fix it if it's a problem. It's also possible to do a control with an organism. This, to me, is the best way of doing it. It's possible to take an anaerobe, do a culture, and put it in your jar. If it grows, the jar is anaerobic. The trouble is, if there's no growth, it could be because the jar wasn't anaerobic or the bug was dead. And because they're anaerobes, they're hard to keep alive, so having a stock bug that's growing is difficult. It's much easier and better to have a bug which is strictly aerobic, like a candida or a pseudomonas, because they can survive on the bench for weeks at a time. Then you just do a subculture, put that plate in the jar. If tomorrow morning it's grown, the jar was not anaerobic. If there's no growth, it was anaerobic. So these are much better controls, and they are just more valid, easier to use. And forget that bit there, not worth worrying about. Talk too much. Running out of time. I can't stress enough that blood agar, anaerobically incubated, is not good enough to go anaerobes. So most good anaerobe plates have extra nice stuff in them to support the growth of anaerobes, which are often fastidious. So vitamin K and hemin and cysteine are really important. And you'll find that most of the basal powder we use to make our plates contains extra cysteine, extra hemin, extra vitamin K. These are all things that help the anaerobes grow. And then we have a range of antibiotics which we use to make our plates selective. A good one is nalidic acid. As a drug, this is a quinolone antibiotic. Most gram-negative bugs that are facultative, like coliforms, are susceptible. So if that drug is in your plate, they won't grow. So you put it in the plate. When you make the agar up, you add the drug to the plate. It's now in the agar. Most gram-pos anaerobes and most gram-neg anaerobes are resistant and they will grow. But Clostridium is often inhibited by it. So these plates are really good for growing gram-pos coccal anaerobes and gram-neg rod anaerobes, not that good for Clostridia. We can use an aminoglycoside, like gentamicin or kanamycin. And most gram-negative rods, like coliforms, are aminoglycoside susceptible. But so are most gram-pos anaerobes, also susceptible. This is a plate designed really to grow gram-neg anaerobes. Or we might use vancomycin. And vancomycin is a drug that kills almost everything that's gram-pos, but nothing that's gram-negative. And we may use these drugs in combination in a single agar plate or by themselves. But these three drugs are the mainstay to make our plates selective for anaerobes. So a common plate is a blood agar plate that contains nalidic acid. They're great because they inhibit coliforms. They're good for gram-negative anaerobes. Great for Bacteroides, great for Porphyromonas, Prevotella. As a side benefit, they're also good for growing staph and streps. Imagine a swab with a staph in it that's crawling with Proteases and E. coli, a horrible wound, messy wound. And then on your mat plate, coliforms are everywhere and you've lost all those nice small pink staphs that are just overgrown. On your blood plate aerobically, no chance. Proteus swarms everywhere. But on this plate, because your gram-neg coliforms have been inhibited, your staph grows by itself. So these are really good at growing anaerobes and really good for growing staphs and streps from mixed specimens. Good plates to use. We can use a Bacteroides biolesculin agar that contains gentamicin. So this is not only selective, it's also differential. Inhibits gram-positives, inhibits coliforms. The bile inhibits most other gram-neg rod anaerobes, and it grows Bacteroides which appear brown. It's a great plate if your question is, does this specimen have B. fragilis? And we can also use a kanamycin, vancomycin laked blood agar if you're really, really fancy. Laked blood means you take the blood and you lyse the cells. And laked blood makes bugs that are pigmented become more pigmented. It's good for growing those anaerobes that are pigmented, which are Porphyromonas and Prevotella. There are two ways to think about anaerobic bacteriology. One way is to say it's really, really hard, and it is, to do it to the nth degree. But to do anaerobes simply is really easy. If all you say is, I'll do the common anaerobes really, really well and leave the rest. That's a reasonable way to do it. Say, all the hard ones, forget it. But the ones that are common, I'll do them really simply. It's like this. The first question is my bug an anaerobe? There are two ways to prove your bug's an anaerobe. One is do a culture anaerobically with a disc of metronidazole. If the bug is sensitive to metronidazole, it is an anaerobe. Lovely method, fantastic. Except there are some anaerobes are inherently MTZ resistant, so it won't work. And there are also some that have acquired the resistance as well, and it won't work then. Not a bad method, but the best method is to take your organism and subculture it twice, once onto blood anaerobic and once aerobic. If the one in air doesn't grow and the one anaerobically does grow, it's an anaerobe. Both ways are okay, just use them with a bit of caution. But you must prove your bug's an anaerobe before anything else. And then, simple tests can help you work out what the bug is. A gram stain is enough, most often, to say your bug is Fusobacterium. Gram-negative rods, big fat bulbous bits on them, that's what it is. End of story, done. 10 seconds. Brick-shaped rods, superfringens. Clonal morphology. Perfringens is big colonies, spreading edge, double zone hemolysis. That's it. If you read at all to do with uni, not just stuff that you read at home, like Women's Day and stuff like that, is that what you read? Oh, not you, Jared, you'd read. What would you read? What's the men's book? Ralph magazine. Is that still around? Oh, haven't you? Oh, Ralph magazine was the man equivalent of Women's Day. It was trashy. I didn't read it. I used to borrow it from it was students who used to bring it in for me. If you read American books, they go on and on about superfringens, and their way of doing it is to use a Nagler plate. A Nagler plate. They call it a Nagler plate. And a Nagler plate is a plate made of egg yolk. And superfringens makes a certain enzyme called a lecithinase that breaks down the fats in egg yolk, and around the colony is a precipitate that goes from being yellow egg to white egg. Not a bad plate to use. And then they take an antitoxin and they do a plate where they put their Clostridium on the plate, and they add one side they have antitoxin that stops lecithinase, and if the bug has got a white precipitate here and not there, it's perfringens. It's a 2-day test at lots of money, and they love that test. In Australia, what we say is, if the bug is a thick brick-shaped rod with double zone hemolysis, it's perfringens. It's much easier and much cheaper. So we don't use Nagler plates because it's a lot of mucking around for no benefit. So I don't use those. If your anaerobe is a gram-negative rod, do these discs. A word of caution, and I can't stress this enough to you. Those discs are really useful. Here I am, here's my plate. The BA plate with nalidic acid in it. And I've grown something. Therefore, it's probably a gram-negative rod. Anaerobe. Gram stain, GNB. I'm on a roll now, feeling great. I now do my subculture, add my discs of kanamycin, vancomycin and colistin. Next day come in, read the zones, go to my chart. Yep, it's a B fragilis. The problem is coliforms that are now resistant and therefore grow on here give the same pattern as this does. So always when you do this test, do at least one more thing. On this plate, as well as your disc that you do for identification, what do you add? What? Spoon of sugar? What do you add? What disc do you add? Add a disc of metronidazole. And then if there's a zone around that one, you know it's an anaerobe. Just makes sense. Or you make your suspension in saline to make your lawn plate to do this, and you put some of your inoculum on this plate and some on a blood plate in air. And then if this grows, you know tomorrow this is not an anaerobe. Don't be sucked into thinking that everything you just do this disc. Always, number one, prove the bug's an anaerobe first, or at the same time. So these are commonly used. Now with multi-toff, all this stuff here has really gone by the wayside. Some labs are still going to use simple tests for anaerobes, but the most complex anaerobe identification would be done by multi-toff. In the past it was GLC and all sorts of funny things. Even by anaerobic API strips, put them in an anaerobic jar and they work. But these days you wouldn't do that, you'd just use multi-toff. So for us, thinking about the practical exam, what I want you to be able to do. If you see a gram-positive coccus that you think is an anaerobe, either just call that an anaerobic GPC or do a disc of SPS. If the bug is a gram-negative rod anaerobe, that is Bacteroides, Physobacterium, Prevotella or Porphyromonas, then do those anaerobe identification discs. If it's Superfringens, the gram stain and the hemolysis is all you have to do. The CAMP test is pretty old-fashioned, forget that for now. If the bug is possibly a Propionibacterium, a gram stain and a spot indole is enough for us here in this lab just to say, probably this. Almost finished. Now, this is important. This is out of a book showing you, for all specimen types, the rate at which you find something compared to finding nothing. First thing to look at is this. Where's the poo? Where is it? Oh, there it is there, stool specimen. Stool specimen, positive, less than 20% of stool specimens are positive. Think of all you know about stool specimens, the pathogens. The Salmonella, Campylobacter, Shigella, Vibrio, Aeromonas, C. difficile, rotavirus, all the funny E. coli's, and there's a massive list. But less than 1 in 5 specimens of stool are positive. That means there are lots of bugs out there that cause diarrhea that we don't know about yet. Or there are things that cause diarrhea that are not infectious. Things like food intolerances that cause diarrhea. Lots of specimens that come in are never ever give you anything positive. Stool culturing is really unrewarding. It's just, honestly, it's boring. Day after day, no pathogens, no pathogens. When you find something, you get excited about it because it's so uncommon. But for today's lecture, what's important is this. Skin swab. Almost always negative. Pus swab, often negative. Wound swab, often negative. Now, if your wound is infected enough to go and see a clinician, or you're in hospital and it's got enough signs of infection for a specimen to be collected, most times a wound specimen that goes to a lab has got a pathogen. And that means we can't find them all these times, all those times, and all those times. I wonder what it is we're missing in these specimens. I know what it is. It's anaerobes, because we're lazy. If you make an effort and you take the time to batch your specimens and culture them on plates that are pre-reduced and put them in the jar very quickly, don't leave them lying around. When you read the specimens, you batch them up and don't have them sitting there doing nothing for all that time. You can change your rate of isolation by, I reckon, double easily. You should be able to bring these numbers down to half of what they are comfortably by doing a few simple things. So, you know, in labs, it's like this. People get old and tired and boring, don't they? In your lab where you work, who are the most senior staff there? John Dados? Is he gone? Oh, he's gone. So what I'm saying is, the older people are boring. They've been there forever. Oh, we do it this way. Oh, another person. They're bored. The time to make a change, seriously, is when you're young and fresh and enthusiastic. When you leave here and get a job, go in there full of energy and tell these old people, this is how we're going to do it from now on. Make a difference, seriously. The time to do it is straight away, early, when you're still keen. After a while, within a couple of years, you'll be dragged down to their level by these boring old people who are sick of it. Get in there and make a difference. Tell them, from now on, we're doing it this way with our anaerobes. And if they're smart, they'll go, great. You're young, full of energy, go for it. Tell them what I said. In the prac, can you remind me to go through this bit? I do it on the board. It's your job to remind me, Tracy. You in the morning group or afternoon group? Who's an afternooner? Your job, remind me. And okay, now the issue is you've grown something. What does it mean? The problem is what you've grown might be a pathogen, might be a coloniser, might be in between. Sometimes, for example, enterococcus in a wound is simply a coloniser. Treat the other bug, it'll go away. But it may be a pathogen, depends on the host. So always the host determines what may or may not be important. And don't forget that quite often what you've grown is a contaminant, either from the laboratory, uncommon, but most often from the patient. Skin flora. Coag-neg staphs in wounds mean nothing. They're just contaminants. Unless the wound is something really weird and the patient is highly immunocompromised. So, an arm wound, I grow a coag-neg staph. I don't care. It's not a pathogen. A wound in someone's chest who's on chemotherapy with an indwelling catheter into a big vein in their chest that grows a coag-neg staph, that's a pathogen. So what you've grown isn't the whole story. It's what you've grown and who you've grown it from and what the body side is. And the wound itself, clean wound or dirty wound. If the wound's a lovely clean wound that's been well looked after, I'm more inclined to think that whatever I grow is important. If the wound is messy and dirty on someone's foot, then it may be just contamination and colonisation. That can be pretty important. As a rule of thumb, when in doubt, send it out. What this means is, unless you are sure and somebody above you in the pecking order says that's a contaminant, get rid of it, don't report it. If you're unsure, report them. You're better off sending a report out saying arm wound, coag-neg staph, unsure of significance, and then the GP makes their own choice about what to do with the result. So unless you're sure, send them out anyway and report them. But with a bit of practice, you start to withdraw some information and only give the stuff that's important. We think that GPs know everything, but they don't. And they'll often go, The lab reported this bug, therefore it must be a pathogen, because you said it was in the report. Their mindset isn't, oh, this is a report, but I'll make my own mind up. They often go, they said it's there, therefore I'll treat it. You'd be amazed how often they treat things that aren't important. Okay, see you in the prac on Wednesday. You're all waiting for me to get out?